Hello, how are you? Your audio isn't working. Yeah. Yeah, I still can't hear you. Nope, can't hear it. I don't know what. Yeah, it's like totally nothing. Yeah. Huh. Yeah, I still don't hear anything. Well,
Hello.
hi. You can try typing in. Hello, Ujjwal?
Hello, hi sir. How are you?
All right. Yeah, I don't know why it's not working on this one, but on this, I don't know if it's the way you have your browser set up or what, because Skype is a different program usually. Skype operates kind of on its own app. So I don't know. We're waiting for Mayukh, and I don't know who else is going to be here today. I know this is a down week for Dick. He comes every other week over the summer. So, okay. Why don't we get started? The meeting. We have a couple things to cover this week. Well, the first thing is updates on GSoC. So, Ujjwal, would you like to present your update?
Yes, so like as discussed, like I have tried to make a workflow for the coming weeks, like how we are going to do this thing and finishing it. So I have, let me share the screen first.
Okay.
Is it visible?
Yeah.
Yeah, so basically, like I was trying and looking for the best like practices of how can we basically integrate our portal in maximum best trade-off that we can get. So like the first like our goal is to host the different models in a very cost-effective manner. So like these are the four principles that we have to keep in mind. Like there should not be any problem with the working; it should be flawless. And our initial purpose that is to impart our models to more and more researchers. This should be fulfilled. We have to like write about the documentation about what library we are making, how to use our functions, and all these things. And also like we have to integrate collab notebooks in that. So majorly, I have divided this project into three major components. Like the first pages are the pages that are your portal for like redirecting flash pages and many other front pages. These all pages are static. Like there is nothing going on. No process is going on here. We just need to render them, and no dynamic processing is happening here. So this is the first part. Second part is the dynamic pages, like where we are actually uploading the dataset and we are actually getting the model back. So this is where all the input taken and we have to produce outputs. These pages cannot be rendered on static servers like GitHub. So we have to render them in some like Heroku, DigitalOcean, AWS, something like that. And then the documentation is a crucial part because we have to tell people like how to use our library and how to install it. We have defined some functions, but how we are taking data from them. So like the every other library in Python, we also have to give documentation for it and the licensing part. So like to make this model very efficient, like we have to mix the approaches. Like we can't only rely on static servers. We can rely on dynamic servers only, but there is no point of hosting static page on dynamic server. They are very like dynamic servers have a very short free range. Like they are not free for very like they are only providing 1 GB of like disk size and there is some downtimes and all these things. So we can't put our all things on dynamic servers and we can't put our all things on static as well. So we have to mix the things. So the plan to improvise overall trade-off is like in order to do this, we need to use like what I am considering is here I need opinion of everyone after like we have given. So basically, I am trying to combine two platforms, GitHub and Heroku apps. I am trying that we can host more and more static pages in GitHub, like on the DevoWorm.github.io. That's already a repository, and you can host all your static pages on there. Like now we need to upload our models in dynamic web app. Like we can use Heroku and different. Like I have taken Heroku as example only. We can use different platforms as well. So like there are two options now. Either we can make a and it has all the models and you can select one of the models and you can run it. So like it's only one portal for all the models. So the pros of these things are like it is easy to use obviously and it gives uniformity. You don't have to go to different websites for the different models and it is easy to develop as well. Like you only have to develop one app which consists of all the models. The cons here is like it may get very big and our free time limit may exceed because free time limit is not so much. Like I have browsed through the codes that Mayukh have read. He has used some PyTorch modules. These all used to be downloaded there in the disk. They are just giving you a disk like a computer which have 1 GB RAM. Like you can think it like that. So they are only giving you this. So you have to download all the like dependencies, all these things in the server itself. So definitely the free limit tag is going to exceed because it is only 1 GB. So this is the con of one of these things. The second con is like, yeah, exactly. Like I have read somewhere, like these companies are usually do tricks like these. As close as we approach to the free tier limit, like if it is 1 GB and we are using 900 MB or something, we are getting very worse performance than what we are getting.
Hello?
Something because of the huge size, but this because of the huge size, but it is something that they do very frequently. Like I have a person who is working at DigitalOcean, like he has also told me like yeah, we do these kind of things. So the second option, what I am considering and what I think is best in this case is like we can make separate app for the different models. Like it can be under the DevoWorm umbrella, but they are different models for the different websites. For example, like if we take Basileia one, so there will be something like Basileia DevoWorm AI.heroku.com, and if you want to go to c.elegance, so c.elegance.heroku.com, and we have a DevoWorm AI touch in all these models. So this is going to be very beneficial in terms of like there is no tension of free limits. As far as I know, like if we are using different apps for different models, it is going to be under the free limit. Like it is a very, very rare chance that it is going to exceed the free limit. And also it is very useful for the coming members and the different contributors to DevoWorm ML. Like we can easily distribute the work. Like for this example, like if there is something called c.elegance, so the person A can be assigned to maintain it. Then there is Basileia. The person B can be assigned to maintain it. So there is no like dependency between these two models. These are separate models. So like in the first point, if we are going to make a single dynamic app, and like as Mayukh and we have discussed last week, we have to improvise our models as long as we are getting new data, maybe weekly, bi-weekly. So there are chances like if something goes wrong, the whole app will go down. But if we are using distributed approach, then if we are working on c.elegance, we are improvising that, our Basileia app is still working, and it has nothing to do with between these two. So this has these major benefits. The cons are like lack of uniformity. Like this is not no longer. This is still under a single umbrella, but no longer a single website. Like you have to go to c.elegance.heroku.com to get the c.elegance part, which will be redirected from the static page. But for the Basileia, it will be the Basileia.heroku.com. Like I don't think so. Like it was very, very big con. But yeah, this is the one point that I can think of. And the second con is like difficult to develop. Like instead of developing only one app, now we have to develop 3-4 apps. Like it is not going to be like difficult word. I don't think like I have put right word difficult. It is not difficult. It is like tiny task. Like it is tiresome task. Like you have to develop same kind of apps for the different models. But again, the pros here outweigh the cons, I guess, because like it is not only good for the as the cost-effective manner, but it's also good for the long-term maintenance as well. And once like these models are evolving, and once if we might get out of the free trial, or like tomorrow, Heroku app says like I'm dismissing my all free trial program, so it will still be effective in the cost. Like I don't know if someone has used AWS. They are EC2 containers. So what people do is they don't buy EC2 containers for the one app. They try to distribute them in the different ways. Like they have to try to make them as distributed as possible, because AWS charges you hourly. So if you are only using 1 or 2 containers, so it might be a case like for 1 hour, you get so much amount of load on the server, and in the next hour you get nothing. And there are also something like your one service is using at very large scale by the users, and one service not using at all. So people try to divide these containers into small groups. So this is from where this approach comes. So yeah. So why Heroku and GitHub pages only? So like I have done some market research in the last week, and I got to know I have like this is the image that I have taken. So like yes, our architecture is fast. We are not service oriented. We are product oriented. So these are. If can you see the table?
Can you zoom in a little bit? All right, thank you.
Yeah. So these are the different platforms that provide us and what kind of service they provide. So only Cocaine is the platform that provides totally free access, but it is like very new. And I have never worked on it, so I can't say like what is the downtime and all these things. I have to like little bit search about it. Heroku app is very famous platform like for the small projects. I have hosted my multiple projects on Heroku app before as well. So this provides free trial for the projects. It is very easy to connect Django and Flask apps on Heroku. And as you can see, like DigitalOcean and all these are charging. And free trial, no, Heroku app doesn't give you any free trial. And for our purpose, we also don't need any free trial because like we have to maintain it for a long time. We are not just going to just see like free trial and we have to like pay because I personally believe like this is the open source initiative and paying for this thing doesn't make sense for us right now actually. Like when we can do it for like free almost. So this is the reason why I'm choosing Heroku. And the GitHub pages, like GitHub pages are the best static servers to host your websites. Like all around the world, people host their portfolios and all these things in GitHub pages. Downtime is very low. I think like it's very rare to get a downtime here. And we can also purchase the domain and directly integrate it to our GitHub page in future. Like if you want to purchase DevoWorm.com, for example, we can very easily integrate the same static website from the GitHub with that domain. Like for now, if we have a domain for DevoWorm.github.io, then the same domain will be shifted to DevoWorm.com or DevoWorm.org, and the same website will be still running. So that's how the GitHub is for the static website, and here we suppose the Django project. So this is the thing that I want to cover here. Like now, I want suggestions from Mayukh and Vinay. So like, if you have any.
Okay, let's see. I have some comments in the comment area here. Vinay said, "Have you looked at Python Anywhere, Ujjwal?" So Python Anywhere.
No, I've never heard about it as well. Let's see.
Actually, it's also a Python. It's also a hosting platform, but it is specific to Python web apps only. You can use Django and Flask and other some other web frameworks like Bottle also. So from what I heard, I think it's completely free over there to have an account to run web apps in Python Anywhere. So actually, the last week I was going through some article, an in-depth review about this Python Anywhere. So which lists all the features like how is it to maintain, how is the platform and performance, and how is the support, the pricing. If in case if we want to extend for a like if in case if our app is like attracting more users and many such things are discussed in the article. So probably you can also look into that just as a second option to see if you find any like advantages over Heroku, then probably you can shift to there. Otherwise, you could continue with the Heroku only.
Yeah, sure. Like I will open the article. I will surely look into it. Thank you.
Sure.
Like the idea is correct, right? Like we can use different platforms for static page and dynamic pages.
Yeah, I think so. I think that's a good strategy. Now I don't know about the for Heroku. I think I'm split on this idea of putting it on under different projects because you end up generating these unwieldy URLs. So it's like you were mentioning, you'd have one for each project, and it would be like, they'd be all different names, and I'm not sure. Well, maybe if we link sort of in the front end to those models, like the models are actually running sort of underneath the surface, and we're able to like, just kind of link to them. You know what I mean? Like I just.
like this is not like they are running on a completely different platform. Like yeah, sorry, they are running on a completely different platform, but still they are in the umbrella of DevoWorm. Like if you want to name the app, you can name app as DevoWorm underscore c.elegance.herokuapp.com, DevoWorm underscore basileria.herokuapp.com. And these all web apps are redirected through through only the static page that the main page of DevoWorm, the face of DevoWorm is. It is only redirected through them. The only point is like instead of having only one app, we have to make an app. Like we have to redirect in any sense. Like even if we are not using Europe or some other platform, we have to make an app in that. So instead, there is 2 points like instead of having only one app, which can cause a little bit overrun on the free tag thing, we can have multiple apps. Still, they can be only under the one umbrella. It is not like that you are operating different apps and people have to remember like which app they have to use. They even don't even notice like they are redirected to different apps.
Okay. Yeah, it might work. I was just curious of how it would play out. I think if we, did like DevoWorm underscore and then the name of the model, I think that would be better. I mean, it would be less, like, people would remember it less, but if we just handled it, like, not at the Heroku level, but at the DevoWorm GitHub level.
Yeah, obviously. All the people are redirected through DevoWorm only.
Yeah. I mean, that's a good idea, I think, to have it hosted like that. We just have to be sure that we know where everything is, where everything's being hosted. Like have an org chart, basically, that we can make sure.
Yes, like for that, like you have so this week I will start deploying all the things on web so that everyone can view it. So you have to like create a new repository for the GitHub pages. Like you can name it as the DevoWorm new, so that I can push all the apps there. So that for the demo purpose, we can see the web page as devworm.github.io/devworm new. So it will show the new website. So that people can start seeing the work. So you have to just create a new repository for it.
Okay. Yeah. Let me know when you want to do that. I don't know if you want to do it like at the end of the project or sooner.
So like according to like what I have planned for the thing is like I will start working this week and I will start wrapping up the project. Like it will take time because we don't only have to make apps and like we also have to make documentation pages, libraries, all these things. So this thing takes a while, so I think I will start deploying at least the static pages on their GitHub straight away from this week.
Okay. Yeah, let me know in Slack when you want to do it. So yeah.
Can you hear me?
Yeah.
Yes.
Yeah. So what I wanted to say was that instead of making a new repository, I think we can make a new branch in the DevoWorm repo. It's devworm.github.io, and you can just test it out on the new branch.
Yeah,
like tested out on new branch, but like to get the people like to get the people like who are not familiar with GitHub as code, so they can just type in the URL and they can see the design pattern. So that's the point that I'm trying to make here.
Right.
Let's see. Okay. Yeah.
So I think Susan, we can hear you now.
Also, Ujjwal?
Can you hear me?
Yeah.
All I have to do is turn off my camera. It interferes.
Oh, okay. Vinay, you were going to say something?
Yeah. So, one last point about Heroku. So, the last time when I was working on Heroku, I saw that the server would there's a threshold time. Like if no requests come for a period of 30 minutes, then the server will sleep for some time. Like if I make continuous requests, like consecutive requests, then the server response time would be fast, because like the server will be up. Like if no user has put a request to the server, then after a period of 30 minutes or 45 minutes, I'm not really sure about the time, I think the server sleeps. So could you just look into, if I'm not sure if that's the case right now, because it's around a year back, maybe the timings have changed, or the way the things have worked changed. So probably you could just look into how the uptime would affect our users and all, because like based on the progress that you're making, I think more people will be visiting visiting the links often. So if the request takes a lot of time, then maybe they'll turn away, there are some chances. So you can just look into it. It's not a very priority issue, I think. And also I request you to look into Python Anywhere as well, the link that I shared. So you can do a high level comparison between Heroku and Python Anywhere, and you can just you can weigh out the pros and cons, and you can take the decision.
Yeah, so basically I tried to open my app, like I have built it some years ago. So yeah, this is something like yeah, so my app is on sleep. It takes like around 15 seconds to open it up. It's a very basic app. So the point is, I think it's still valid. I will try to open it up again in few minutes to check like what is the downtime. So yes, we can. I will obviously look at the Python Anywhere. I am not aware about it. I have just to study it. I will definitely look into it. And I think like there is a tweak we can do for the downtime if it still exists. We can make fast requests on the server like in no time, like on every 40 to 50 minutes we can just hit the IP address so it will not sleep. We can do something like that.
Oh, so you're trying to automate the process of hitting so that it won't sleep, right? Is that what you're telling? Yeah, even that can be done, but I would suggest you to look into their doc somewhere if it's mentioned, if there's an option to avoid that thing, avoid doing that thing. If there's no such option, then probably you could think about hitting the API every few minutes or something like that.
Yeah, so what I will do is I will first study about the Python Anywhere, what you have given, and if I think it is better, I will obviously switch it to it. And if I think some advantages are there on Heroku, so I will definitely look into it and report it in the next issue.
Yeah, and please also consider the learning time that it would take, the learning curve it would take into putting everything into Python Anywhere, because you don't want to get stuck at some setting issue, right? So once you feel that it's easy to use and easy to get started, only then you can make a shift.
Yeah, okay, sure. Thanks.
Yeah.
Thank you, Ujjwal. That was good. We'll keep on talking about this in Slack. Mayukh, do you want to give your update?
Yeah, I'll just start with my update then. Wait, let me just share my screen.
Okay.
Yeah, can you see it?
Yes.
Yeah, so what happened is that last week I didn't, I'll start with this one. What happened was that the data was not clean, okay? So that was kind of my objective last week, to find out the points where it was somehow corrupt. So that is what I found. I found that in some of those videos annotations, basically the populations go up and up and up, and then they suddenly just fall to zero or some kind of a random value, and then they just give that, which is not right for that specific frame, because the population of the cells are supposed to increase, right? But they were not increasing. So I found these using some simple NumPy algorithms, and then I just removed these out. And then before removing, there were these kind of cliffs. I call them cliffs because they're these things, right? So I just removed these cliff tags. Tags are basically just file names. And then I just trained it again on these clean videos and clean frames. And I'll just go remember last week it was not predicting well on the early part.
Yeah.
So now in that you can see it, right? It's predicting much better.
Okay.
Yeah, I also tested it out on a lot more file names, so you can see like it's much better than before. Like sometimes it's failing, I don't know, because like some noise or something, but it is pretty accurate. We have to assume that there could be human error as well when like manually labeling all of them. So if we consider all of that, it's hovering pretty well around the original values. Like you can see it, right?
Yeah.
Should I zoom in?
Yeah, zoom in a little bit.
Yeah, so this is working pretty well. And other than that, I mentioned in my proposal that I'll also start working on this kind of a small Python library which the users would be able to use for for accessing the models. Like either like someone who is like an endpoint researcher, he would like to use the website. But if someone wants to scale it up and do it on a lot of stuff, like on a lot of videos or on a lot of images, and if he or she knows like Python, then instead of the website, they can also use this small Python library that I'm building. I'll just go into that. Yeah, so the code is basically, it's all explained on the notebooks itself, so I won't go into that. I'll just show you the README and how it affects. Like instead of this, a lot of like a gob, like instead of using gobs of code to just import the model and then run and find these kinds of plots, I just wrote some, like I just shortened it out, and it's already there. I'll just send the URL. Wait, I'll just send the URL into the chat. Yeah, this one. The name is just temporary. It's just a code name kind of a thing. We can just change the name and everything later on. But for now, what it does is that we can just, the user can just simply import, like from PyElegance, import lineage population model. And the lineage population model is basically this model that I'm talking about. It can look at videos or frames and then predict the population of the cells. So we can just import it, like it would be this simple, like from PyElegance import lineage population model. The model equals to, we just call a class lineage population model, mode is CPU, like CPU or GPU. Like if someone has a GPU, they can use it for faster predictions. But for now, like for me locally, I'm using it on a CPU. And then we can just prediction equals to model.predict path sample.png. Like we can just give it an image path, and it can just return me a list of values of the, not a list, it gives me a dictionary. Like A is this population, B is this population, C is this population, or like those cells, right? And then also we can use it to predict on videos, like model.predict from video path. And then we can, like, since it's a video, we generally don't like to take a very big data frame into the variable and just work with it. Instead, what we would want is to save the corresponding results of each frame into a CSV. So that's what I did. You can just hit save CSV equals to true, CSV name is equal to some file name. So you can save your CSV files that way. And other than that, you can also directly plot the model's predictions from the video into a plot like this. So basically using this one function, I can just, I'm shortening, like, wait, I'll just show you. I'm just shortening all of this part, like all of this part is shortened, and like you can just use one function to go from like the video's name to a plot like this, which can be saved as a photo. And you can also do some stuff with it because it's a matplotlib image. Yeah, so that's pretty much what I did.
So like I have seen this repository before, and like this is the work I'm talking about. Like we have to do documentation. It is a very pretty good documentation right now here. But like instead of having a documentation on GitHub.io, we should definitely transfer it to the website. And also like right now it is at PyElegance.
Name I gave. Never mind the name.
Yeah. So like what I want is like we should transfer it to the pip install so that people don't have to come.
That's my final goal. Like to make it into a complete package. That's what
so rather than having a different name for C.elegance and for like Basileia and some other models, what we can do is we can, like, what I have planned is we can create a pip install DevoWorm AI, DevoWorm ML, and under it you can have the PyElegance Basileia models, like from so instead of from PyElegance, it will be from DevoWorm.
PyElegance.
Exactly.
Yeah, I get it. Yeah,
because
so there would be digital Basileia on one side and PyElegance on another side. So both of these will be under, I get it. Yeah. So we can publish
And also
DevoWorm.
Vinay Vias model like on the segmentation of the image of speed. All these models under
Yeah,
DevoWorm.
yeah, that one would take time, I guess, because that's not PyTorch, that's Keras, and it's quite out of my domain. Because I spent less time with Keras than I could see, or we'll look for help maybe. Yeah.
Yeah, this is great. Yeah, this is obviously like a code name, or internal name, but
This is made for like people who are not like great with Python. Even they can use it. Like researchers who just learned Python just to research, like just to plot stuff and just to see. Like those who know researchers who know basic Python, they can also use it.
yeah.
So that's how I'm trying. Like it's very simple and it's kind of flexible. You can save the CSV or not. Like you can return it. It even returns a data frame, so you can work with the data frame. If someone knows Pandas, which is a very famous data science library, they can use the data from the model and then they can do stuff with it. So it's kind of very like it is made on top of a lot of data science friendly libraries, which I've been or which are like worldwide. NumPy, Pandas, SciPy. So it's pretty like for me it's like it's going well for now. I'll add the other models.
Mike, you can do one thing like when I was browsing through the repository, through the code, you can add a help function as well, where you can like give all these functions. Like someone has typed help, and he can get all the how to use these all things. Like these are really like not a big task. Like you just have to print few statements about all these things in the library itself. Like there's a basic like you can say
Could you repeat?
procedure or some like help function. You can make a help function. Am I audible?
No, this is not exactly a like what I can say is that this is not actually some command line app. Like in command line app we have dash h, right? Are you talking about something?
Like not that. No. I'm talking about like so if someone has imported the library,
Then dot help. I is dot help. Then it will print the whole thing.
he can use like there's a function, for example, lineage population model. So you can have a help and double bracket or double close curve brackets. So he can get all these commands on the it's Jupyter notebook as well. So he don't have to like cruise through website like oh yeah I have to do this, I have to just go there. He can just have like this is something like bit of like improvement. It is not necessarily at all. But, like,
Yeah, I get
it is.
Actually, you know what's going on. Actually, I have my exam soon tomorrow, so I'm pretty tight right now on my schedule. So I'll start with these things.
You don't have to do it right now. Like it can be done later in the week. Like, I'm just suggesting.
Issue. I think someone can raise this issue, like on the GitHub. So I'll remember it. Otherwise, I'll just lose track of it.
Okay, what was the issue?
To add a help section in PyElegance. For now, it's the code name. So yeah.
Yeah.
Yeah. All right.
I think that's a good place to stop. Yeah, this is a good conversation. We'll have to keep going in Slack. And yes, this is a very good tutorial sort of introduction to it. So that looks very good. And Susan said that it would be very helpful to her as a beginning Python user. So this is a gift someone already who finds it to be useful. So that's good. Thank you for the update. I'm gonna. Can I share my screen? Thanks. Yeah. So now we'll move on to some other things here. So why don't we go first to the board? I was gonna go to that anyways, but we're gonna go to it now. So you can see my screen. So this is the board again for GSoC 2020. We go into the projects, the project board, go into this, and we'll create an issue we just talked about for how do we create a project here? Or an issue. For some reason I can't do it. Oh, did they change this or what? I don't know why I can't seem to create an issue here. Let's see a new issue pops up. Okay, we'll just do it this way. So this is and help section. Oh, I see why. I'm in the wrong, logged into the wrong. So maybe I'll do this later. I'm in the wrong account. I switch between accounts, and that's why I get, like, sometimes I can't do things. But that's okay, I'll do it later.
Yeah, we will take it to mind.
Yeah. So why don't we go back to the board then? So we have this is to do. I don't think we've started on that. Krishna.
No. Actually, I have started on that. Like on the ODC thing, I have started on the introduction material. You can put it in progress.
Okay, we'll put that in progress. Well, it's not even letting me move the things around. Huh. Well, okay, I'll put that in progress later.
Yeah, I think we can move that one to done. Review Mayukh's graph on predicting cell divisions during embryogenesis.
Alright.
I think we can move it to, because we just reviewed it today, right?
Yeah. Which issue was that?
Review Mayukh's graph on predicting cell divisions.
39.
Yeah, this one's done, right?
Yeah. Alright. Optimize color palette. I think we're done with that.
I think, yeah.
Yeah. Alright. Create pages, reorganize. Number 17.
I have created the pages for DevoWorm and Devo methods.
Okay. So we can probably just put that in done or in progress, do you think?
Audio? An audio?
Yes.
Yeah, so we can keep that in progress.
Okay.
These are the kind of things they have always have the scope of improvement.
Yeah. Work through Epic dataset. That's in progress, I guess, because we're still kind of working with the data. And then design evaluation, that's still open, but I think we've kind of gotten some feedback on that. I'm not sure what we actually have in the comment here. I can't remember where we were putting that stuff, but I've gotten a little bit of feedback, but I guess I've communicated it to you. Let's see. Static hosting number 40, we were talking about this today, so that's sort of in progress. I'll move that to in progress. And then this one here, number 7, that's the one, the movement tracking and prediction. That's still on hold. Integrating models with web portal. We were talking about that as well as part of 40. So we'll move that to in progress. And then integrate cell tracking with segmentation. That's a bit down the road. So I think that's good. Like I said, Krishna is still open for a presentation. I've talked to him. He's still working on it, I guess. So that's good. That's the GSoC board. Now I wanted to talk about this periodicity paper. So I think Ujjwal brought it up last meeting, or I think we talked about it last meeting. This periodicity paper that is, if I go to this link here, I think I can pull up the outline. This is an extended abstract that we never really got off the ground, but we're going to do it for a special issue here. So this is emergence of order in space, diffusion of order in time. And Susan might actually be interested in this too. This is the idea of tempo and mode, which they've used in the context of evolutionary biology. But what we're also looking for is to look at that question in the context of development. And so it's, there's a mix of computation and data science and sort of biological interpretation here. So, you know, there are a couple. If you read the extended abstract, why don't I put it in the chat so people can read it, and we can. You can tell me if you're interested or if you have ideas for contributing to this or not. And so it just basically it's finding these patterns in different types of embryos, these temporal patterns. So in zebrafish during early embryogenesis, and this is the zygote stage, this is the cleavage stage, you have these pulses of cell divisions, or these rounds of cell divisions in time. And you can see it towards the end of the cleavage stage, it kind of breaks apart and becomes asynchronous. You see some of that in C. elegans as well, with these patterns of division, these rounds of division. And you even see it in the post-embryonic worm, where you have cells that are differentiating. And so there's that pattern. So that's the idea behind the paper. Now what I've done this week is I've come up with an outline for sort of a path forward on this. So this is the to-do study plan. So there's an objective. We have participants. I can add Susan if she would like. A division of labor, so we're going to divide up the labor. I think Mayukh, his division events analysis, we could maybe use some of that in this paper. We could just, you know, if you want to generate a couple of graphs from the data, we can maybe see some of these. We'd have to transform the data a little bit differently than what you have there. You know, one way to show this effect is to use histograms with bins of different sizes. I put 50 minutes there. That's sort of arbitrary, but I think extracting the data directly from images is an interesting data set to use, because it might confirm what we've seen in some of the nucleus tracking data. And then Ujjwal, you know, you can do a number of things: data analysis, frequency analysis, as you mentioned. I'm kind of coordinating this, and Jesse and Dick are interested in manuscript review and probably other things. And then Susan, I can put you in for something if you want.
Okay.
Yeah. The data sets are, right now we have C. elegans. So we have C. elegans, kind of this division of 450 minutes of embryogenesis. We have, and this is an example. This is the control here of the simulated embryo, which I've not shown to the group, but it's this this program that, you know, you just basically take the idea of a dividing, you know, cells dividing, and you do it at different, according to different distributions, and you generate these data that are simulated, and I call that the simulated embryo. And that we use that as a control to compare that to C. elegans and zebrafish. And so we have the zebrafish data up to 450 minutes as well, and that's a different data set we can go over. There's this ZFin site which actually goes through the different stages of zebrafish development. So if you're interested in learning about zebrafish development, they have all of this sort of laid out. So zebrafish development is different from C. elegans development. You have the zygote and the cleavage and the blastula, gastrula, segmentation stage, pharyngula stage, hatching stage. So this is at a bit different time scale than C. elegans, and it's a bit more complex than C. elegans. But these data exist. We have the nucleus tracking data for this that we've gotten from one of our partners in data acquisition. And then so this is like a canonical sort of accounting of cell division here. You can see that, like, you get into the at the end of the cleavage stage, you have 128 cells. So, you know, we have a bit beyond that, but we'll, you know, we can kind of hash that out as we go through this. And so there were some ideas about like looking at the difference between C. elegans and zebrafish data. You might be able to simulate those data then to maybe shift the shape and timing of cell division to see what happens. I know that's a little bit odd as a concept, but basically taking the existing data and sort of applying some sort of distortion to it to see what happens to cell division. If you do that, and I mean, I'm just thinking of like ways you can do like little computational experiments with the data, but that might show something as well. We might learn something from that as well. So Vinay, thanks for attending. I know he left. So yeah, thanks for attending, Vinay. And he said, great work, Ujjwal and Mayukh. So that's good. Yeah, so that's the, let me see. That's our, this periodicity of the embryo paper. And we'll be working on this for a while because we're trying to target a special issue of a journal next year. That's the first draft is due next year, so in 2021. So we have a bit of time to work on this. But I'll be bringing it up in the meetings, and we'll see how much progress we can make on it. So if you have any, ideas about it, let me know. Next thing I wanted to talk about was that I attended a conference.
So the link, could you just send the link of that study plan?
Group meetings? Yeah.
Yeah.
So this is the link for the group meeting.
I also have a question, like in this ZFIN data set, we have different processes, like biological processes going on at different times, right? So we are trying to find a pattern in all these processes, or like they can be different pattern in these different processes.
Well, yeah, we're just looking at the periodicity, like, of cell division. So, like I showed you the graph with the zebrafish, you have this strong sort of cyclical behavior. I don't know if I have to go back all the way to that. But then it breaks down after a while. And the idea would be to like figure out where in developmental time these cycles are happening and maybe trace it back to the embryo, or, do some sort of simulation that shows that, there's some importance to the, well, we actually do have the XYZ coordinates of these data too, so it's not like we can't go back to that and look at like what's going on in the embryo in terms of the cell positions. So we have these in zebrafish here. So the idea of the zebrafish, the ZFIN page I showed you was a stub that just basically described the progression of development. So on C. elegans, you really don't have too many stages within the embryo. You have like a comma stage and a pretzel stage and some other stages, but in zebrafish, you actually have very distinct phases of sort of how the embryo exists. And so this is the zygote stage here. This is the cleavage stage. And then there's this sort of breakdown in this sort of periodicity of cell division. So we might ask why that is. And, maybe it's that it's getting, we get enough cells, or maybe it's starting to develop tissues that just, account for this sort of change in regime here. And so this is a very qualitative account. But what we want to maybe do is link it back to the data, to the anatomy, and then also maybe describe this a little bit more quantitatively.
So like, if I'm getting right, we have different kind of data set, and we have to just find out like if there is some sort of periodicity in these data set. What is happening there? Like, for example, like what Mayukh has done with the C.O, epic data set, like there is some sort of periodicity, like it is rising up and then flat, rising up then flat.
It doubles after
kind of thing that we are aiming.
Right.
It doubles basically after a certain interval. That's what I observed.
Yeah. After it's at different intervals you have these different patterns. So like what Mayukh showed was like sort of a cumulative increase in the number of cells, and you can see that pattern where it kind of levels off and then it comes back up. And if you express that as a histogram, you'll see this pattern of a cyclical pattern. But then, the question is, how do you interpret, say, the period of that? So the period between peaks, is that the same across development? Does it decrease, increase? Is that something, because there's a period of time between cell divisions, right? There are rounds of cell division. You have them at 10 or 15 minutes, maybe 20 minutes. And they're not necessarily regular over every set of cell divisions. But there are these also, apparently there are these sort of time, things that happen in a certain period of time, and then there's no cell division, and then there's another peak of cell division, and there's a period between those two waves. And so the question is what is this period in between here, for example, what is the significance of that? That's, above and beyond like individual cell divisions. This is like the sort of the mode of this developmental process. Remember, this is all occurring on a tree of cells differentiating. So they're differentiating on this lineage tree, and we actually talk a lot about differentiation trees, and so there might be a way to link that in too, because differentiation trees are a little bit different than lineage trees. They describe sort of tissues and their relative to one another. So there might be something there too. So I put the links.
So
go ahead.
I think we can, me and Mayukh can definitely help to find out if there exists some pattern. But the interpretation part is it is not something that we are aware of. We don't know about biology and all these things. So,
well, yeah,
we can definitely look into the patterns if there is.
Yeah, interpretation. Yeah, that wouldn't be your domain. So that's why I split the division of labor up. So some people were some people were interested in maybe expert at analyzing data. Some people were expert at thinking about embryos. So we have both sides there. So I put the links in the chat. So why don't I move on to this thing. So this week I went to a conference called Artificial Life, and I had a chance to do an impromptu presentation on OpenWorm. And so I was introducing OpenWorm to this group of people who may or may not have heard about it. I'm going to go through, it's a 5-minute talk, so I'm going to go through it really quickly and see what you guys think. So this is a group of people, they do simulations of life, simulations of evolution, and some development. Some of them have heard of it, of course, some of them not, but I'm going to go through this and, as if I were presenting to some educated group who was interested in knowing more about OpenWorm. So this is building the world's first virtual C. elegans. And so I mentioned that it's open source, and then there are all these contacts that ways you can access the foundation, the different projects. So the clickbait version, of course, is this Lego robot that you sometimes see in news stories. It's like scientists put a worm brain in a Lego robot, and you won't believe what happened next. And so there's a lot of hype around this, which isn't even really the state of the art here. But what's going on is this robot is controlled by these neurons, and this is basically C. elegans Connectome. So C. elegans Connectome is 302 neurons, and we know the connections between them. And that's not true of every connectome. So it's a unique situation with C. elegans. And because we know all the connections, we can reconstruct the connectome relative to behavior. So all they did was simulate this connectome and use that to drive the robot. But this is Stephen Larson. He's one of the founders, or one of the first people who started OpenWorm. And there's a scientific question here, and they presented on this sort of thing at NeurIPS in 2017 and the Royal Society in 2018. And the question at that time, and it still holds true, I think, was that by simulating a small nervous system, can we learn information processing principles about how it works that we can apply to the human nervous system? And so we can refine that question by asking about organizational principles. And we do this in the OpenWorm Foundation. We have a bunch of projects that fall into these, that explore various aspects of this. So we have the OpenWorm Movement Database, which is a database of movement data. So if you want to understand how the worm is moving around and behaving, we have data on that. We have sort of next generation robots. So this actually looks like a worm, and they're using the same principle to drive the worm, but it's a bit more refined. So we have that going on. We have WormSim, which is a program that allows you to simulate the nervous system. Geppetto, which is similar to WormSim. And these are models that plug into like platforms like Neuron. So you have a good basis for reusing the models and making them more relevant to, the computational neuroscience communities. You have Cybernetic, which is a software that simulates the biophysics of the worm. So it's movement, but it's the biophysics of the movement, not the behavior. And then DevoWorm, which we explore development. You know about that. So this is something I pointed to sort of tongue-in-cheek. We have so many things going on that someone commented, it looks like the complexity of the dependency graph between the GitHub repos of the OpenWorm project will soon be comparable to that of the nervous system of the actual worm. And so this is in the OpenWorm GitHub repository. You have all these different dependencies, and that's the idea. But that's what happens in an open source project, and when you're trying to do something like this. So by simulating a small embodied nervous system, can we learn about its information processing and organizational principles, and we can similarly think about emergence. And so this is the idea behind OpenWorm. In one sense, is the model realism. And so you can see in this case you have two frogs and two airplanes. One is a real frog and a real airplane. The other is a model. But the models are realistic. So they're hyper-realistic and not abstract models. And so on the right you can see an example of the OpenWorm model. This is sort of the inside of the worm, the muscle walls and the axons leading to motor neurons. And so this is very hyper-realistic. And I would recommend this special issue of the Royal Society Journal. There are a bunch of papers in there on OpenWorm and the various ways in which you can model worms. And it's really a good exposition of what's going on in OpenWorm. And so Matteo Cantarelli, who's another person who founded OpenWorm, he talked about at the Royal Society meeting they had this idea about a Turing test for worm movement. And so a Turing test, of course, is something where you test a computer to see if it's, they do it in AI for different programs to make sure that they're not generating randomness. This is something you can do for a worm phenotype. So one of these is a simulation. The other is an actual worm crawling out an agar plate. And so he says, can you tell which is which? And of course, the idea is maybe you can't. But that's the idea. We don't have a Turing test for worms, but this is something that's a really fascinating idea. When you have these realistic simulations, you can actually ask questions like this. The problem, though, is that you have this hyper-realism. So one option is to have a spherical adult worm in a vacuum. So you might have like a hyper abstract model of the worm that might give you like biological relevance, but it's not very realistic. On the other hand, you have these hyper-realistic models, but they actually don't a lot of times account for things like adaptability, robustness, and evolvability that we might expect out of a living system. So that's a problem. But nevertheless, we have these nice models. So they come from, so this is a classic paper by White in 1986. and they explored the anatomy of the nerve ring. And so they're looking at, these are anatomical diagrams of the nerve ring and its network. So there are a bunch of neurons here. They're defined with names, nomenclature names, and they have connections. And you can figure out all the connections. Like I said, it's not a very big connectome. And you have these diagrams at different, anatomical orientations. And so you can take that sort of data and produce something like this, which is a worm. This is a worm viewer view of, this is from browser openworm.org. So you can play with this online. And this is the sort of the muscles, and you can add muscle wall, and you can add cuticle, and you could actually have a full worm, or you can take away layers and have this minimal representation of the worm. But it's hyper realistic in that it's all sort of in place. And this is the adult worm, so this is not development. So you can visualize the 302 cell connectome. So why would you want to have a hyper realistic model based on real data? Well, one reason is you want to build these type of models and, simulate behaviors. But you also want to visualize the data and have these find these new relationships. So in this case, we have sensory network of sensory neurons and a sensory network of motor neurons. These are I think the chemical synapses, and these are the electrical synapses, or the gap junctions. And so those two networks give you different results if you look at them. The gap junctions are electrical connections that form directly between cells. And so those are actually different than the chemical synapses, which is what we think of when we think of Hebbian learning and action potentials and things like that. Gap junctions can do some of those things, but it's a different mechanism. And so we can ask things like, how are gap junctions and synaptic components of a connectome generated in development? And so there's this paper that's coming out in Frontiers in Cellular Neuroscience. It's something I've been working on. It's this model of connectogenesis. So I'll send this out to the group when it's out. But, there's also this thing about single cell simulation of ion channels. So for each cell, we know the cell name, and we know kind of how it's connected. But we can also model within the cell. We can model these ion channels as blocks. So we have models where we model different components of flow of ions in these channels. And so this is the kind of thing that allows a cell to become depolarized and create an action potential. But we've treated all this as blocks. This is a series of blocks and computational elements. And then there's just basically a directionality to this. So, this is a very stripped down version of it. But this is basically what we can do. And so we can actually simulate this at multiple scales. So we can simulate ion channels at the single cell level. And we can add those cells into networks and manipulate all of it in a sort of a connectome model. And so this is C302, which is another project. And it allows you to simulate a lot of the connectivity and the dynamics of it. So that's it. That's the whole talk. And then I guide them to this Docker simulation. So this is the Docker container I've talked about. This is actually available on the OpenWorm GitHub, and you can download it, and it's you can use it in Linux. There's no Windows version yet, and then you can visit all of our little outlets, media outlets, GitHub, Twitter, YouTube, and join Slack. And then these are the senior contributors, and there are a lot of other contributors. And I mentioned that it's like hundreds of people working towards this goal, and then I encouraged people to join in. So that was the talk that I gave to this group of people. It was pretty well received. So let me look at the comments. Oh, could you post your presentation with some of your word explanation? Sure. This talk will be on YouTube, and then I'll send out the links to the slides. And then you can use chaos theory for a type of Turing test. So yeah, you could use that. Yeah, you could look at how the people looked at, we have movement data. So I know that Mayukh has been looking at the movement data and, kind of simulating it, but it exists in this database. We can actually decompose some of the movement. The movements are pretty stereotyped in C. elegans, but I know people have done looked at other organisms, looked at traveling patterns, and there are a lot of theories about levee flights and things like that happen in movement of organisms. So they explore a little bit of space and then they move on. They take a transit to another place and then they explore, they transit to another place. And there are a lot of, ways you can characterize that with chaos. So it's not, it's a pretty good idea. So yeah, that's good. So I guess we're over time here. Thank you for meeting this week. Oh, okay. Susan had another comment. Fighting fish looking in a mirror with deterministic movement equals aggressive movement. Yeah. Do you want to explain that a little bit more? Are you there? Chaos theory.
I guess you can't hear me.
Oh, I can hear you now. Yeah.
Well, can you hear me?
Yeah.
Okay. Well, it was chaos theory was used to determine whether it was random movement or deterministic movement, and fighting fish in a mirror definitely has deterministic movement toward its image.
Yeah.
So there was a paper written on this. So, you can definitely use some sort of fractal or chaos theory, and I forget what it was. It was a while ago, but you can definitely use it to figure out whether movement is a guided movement or whether it's random.
Yeah. Yeah, it's that would be an interesting analogy. I don't think anyone's written that paper. I mean, C. elegans, we think they're stereotype behaviors. So we observe different movements, but they do a lot of other intermediary behaviors that are not well explored. They have quiescent modes, and they have they encounter maybe a stimulus and back up, and then maybe make turns. But other than that, there are all sorts of things that we don't really. I don't think people have. I think there are a lot of behaviors that aren't characterized. How's that? So.
Oh well, that would probably scope for a paper.
Yeah, that'd be good. Yeah, if you find something, that would be good.
I'll try to find the fighting fish paper for you.
Okay, that'd be good. I can send it to the rest of the group, if you want to present it, you're welcome to.
Okay, I'll see how much time I have.
Yeah.
I keep getting, he said, well, I want you to read this, I want you to read that, I want you to read the other thing.
Yeah, it's very challenging. The reading list always grows. That's why we're doing the reading queue that I didn't get to today, but that's, always something interesting, though. So thanks for attending today. I think everyone's doing really well. And hope you're staying safe, and we'll talk next week. Next week we have updates on GSoC, and then maybe we'll have some presentations, Krishna and maybe some other people. So have a good week.
Bye.
All right, bye. Good luck in your exams, Mayukh.
Yes.